guys, hope your spring break is going well. I just wanted to make a video here going over the answers to homework number 9. I know I missed a chapter that kind of was loaded up with information, so hopefully the sub explained this all to you guys and you understand sort of what's going on here in this chapter about genetics. So feel free to follow along and do the questions with me. If you've already done the questions, feel free to check your answers. But this is some important stuff that's going to pop up in the chapter quiz. So I want to make sure that you guys have a decent understanding of how to solve these problems. Okay, so let's just jump right in. Question number 1 here. So in rabbits, short hair, capital H, is dominant to long hair, lowercase h. So part A, if a rabbit that is homozygous for short hair, so homozygous meaning you have two of the same allele, one from mom, one from dad. So homozygous for short hair is going to be capital H, capital H. If that is crossed with a rabbit that is homozygous for long hair, so homozygous, again, two of the same allele, and this is long hair, this is going to be our homozygous recessive individual here. We're looking for the genotypes and phenotypes of the F1 offspring. So this is our parental generation. When we cross those, the result of those crosses is going to be our F1 offspring. So we can figure this out by doing a Punnett square. So we're going to draw our Punnett square here. Across the top, we're going to put the possible alleles from one parent, and then down the side, we're going to put the possible alleles from the other parent. So our homozygous dominant individual could either give a capital H, or they could give another capital H. And then down here, our homozygous recessive individual can either give a lowercase h or a lowercase h, right? They have two of the same alleles. That's their only option for giving alleles. So then let's populate this Punnett square. We're just going to combine these alleles to see what the possible, that's B, what the possible combinations are in this generation. So the possible genotypes, they're all the same. All of our individuals here, all of their genotypes are going to be heterozygous, meaning they have one of each type of allele. So one dominant, one recessive, a capital H and a lowercase h. And then the phenotypes, when we have a phenotype, that is the physical characteristic of the genetic makeup. So we're going to look at our genotype here to determine what our phenotype is. Anytime we have a heterozygous individual where we have this complete dominance situation where there's not something else kind of going on, and you can check the PowerPoint to see sort of the examples of when this isn't a complete dominant situation. But typically when we see this, the phenotype is just going to be this dominant allele. This recessive allele is going to be masked. So the phenotype is going to be based off of this capital H, which is short hair. So all of these individuals are going to have short hair. Part B, if two of the F1 rabbits are crossed, what genotypic and phenotypic ratios would you expect among the F2 offspring? So F1 offspring were the result of our parental generation. F2 offspring are going to be the result of an F1 generation. So we're going to take a heterozygous individual, cross it with another heterozygous individual. Let's make our Punnett square again. So our heterozygous individuals can give either a capital H or a lowercase h. So let's populate this Punnett square here with all of the possible genotype combinations. So the genotypic ratio is going to be 1 homozygous dominant to 2 heterozygous individuals to 1 homozygous recessive individual. So a 1 to 2 to 1 ratio, genotypic ratio there. And then our phenotypic ratio, so 3 of our individuals are going to have these capital H's, so they have short hair. 1 of our individuals has 2 little h's, so those are going to be long hair. So we're going to have 3 short hair to 1 long-haired individual. So at a 3 to 1 phenotypic ratio there. All right, so in turtles, question number 2 here, in turtles, green shell color G is dominant to brown shell color, lowercase g. So a turtle that is heterozygous for green shell color, heterozygous, 1 big G, 1 little g, crossed with a turtle that is has a brown shell color. So if you have the brown shell color, you have to have 2 little g's. right? You have to have two recessive alleles to see the recessive phenotype there. So what is the genotypic and phenotypic ratios here in the offspring? So let's make our Punnett square. So our first individual can either give a big G or a little g. Our other individual can only give little g's. So let's see what combinations we have here in the Punnett square. So our genotypic ratio, we've got two individuals that are heterozygous, and we have two individuals that are homozygous recessive. So we can just say that that is a 1-to-1 ratio, right? For every one heterozygous individual, we have a recessive individual, also a 1-to-1 genotypic ratio. And then our phenotypic ratio is going to be, so we've got two individuals here that are going to have green shell color, two individuals here that are going to have brown shell color. So our phenotypic ratio is going to be very similar to our genotypic. We're going to have 1 green to 1 brown, so a 1-to-1 genotypic ratio there, or phenotypic ratio and genotypic, right? 1-to-1 for both of those. All right. Question number 3. Question number 3 here. There we go. In humans, sickle cell anemia is a disease caused by a mutation of a single locus gene, which codes for an important blood protein. The allele for normal hemoglobin protein, capital H, is dominant to that for the one causing sickle cell anemia. So sickle cell anemia is a lowercase h there. So what alleles would be produced by an individual who suffers from sickle cell anemia? So if you are suffering from sickle cell anemia, you have to have both of those recessive alleles. So you are going to be homozygous recessive. And so all of the alleles that you produce are going to be little h's, right? You only have little h's. Those are the only ones you can produce. So you're going to just be producing those recessive alleles. Part B, if an individual with sickle cell anemia and an individual that is a carrier for the disease have a child, what is the probability this child will have sickle cell anemia? So we have an individual who has sickle cell. We are crossing that with a carrier for the disease. So if you are a carrier, you don't have it, right? You have the dominant phenotype, so you don't have the disease, but you are carrying this little h, and you can possibly pass it along to your offspring. So we have one big H and one little h there for our carrier. So we're looking for the probability that a child will have sickle cell. So we're going to make our Punnett square. So it doesn't matter if you put which parent you put on the top and which parent you put on the side. I personally like to do whichever one I write first in my cross, I put across the top. That's how I do it. But again, it doesn't really matter as long as you have one parent going across the top and one parent going down the side, you'll be all set. So if we combine all of these alleles for our possible genotypes of the offspring, so what's the probability that they will have sickle cell anemia? So if you have sickle cell, you're going to have two little h's. So that is 50% of our alleles here, right? 2 out of our 4. So there is a 50% chance that the offspring could possibly have sickle cell anemia. Okay? All right, question number 4. Let's move along to question number 4 here. In pepper plants, green fruit color, capital G, is dominant to red, little g. So if two heterozygous plants are crossed, what are the expected genotypic and phenotypic ratios of the offspring? So we've got two heterozygous plants here, big G little g times big G little g. So let's cross those in our Punnett square. Big G little g, big G little g. So we're going to populate it, fill in all of our possible combinations of alleles. So our genotypic ratio is going to be 1 homozygous dominant to 2 heterozygous individuals to 1 homozygous recessive individual, right? 1 to 2 to 1 genotypic ratio. Our phenotypic ratio here, 3 of our individuals have these capital G's, so those are all going to be green. So we've got 3 green. And 1 of our individuals has 2 little g's, so those are going to be red. So we've got a 3 to 1 phenotypic ratio here in this cross. So you'll see this phenotypic ratio pop up pretty frequently. Anytime we're crossing two heterozygous individuals, we're going to see this 3-to-1 phenotypic ratio, right? This 3 individuals that have the dominant phenotype, 1 individual that has the recessive phenotype. Okay, question number 5. So, wood rats are medium-sized rodents with lots of interesting behaviors. You may know them as pack rats. So we're going to assume that the trait of bringing home shiny objects, capital B, is controlled by a single locus gene that is dominant to the trait of carrying home dull objects, lowercase b. So 2 heterozygous individuals are crossed. So 2 heterozygous individuals are going to be crossed here. So in part A, we're looking for how many of each genotype are expected, and in part B, we're looking for how many of each phenotype. So this is just like what we did before in question number 4. So we're going to make our Punnett square here, our heterozygous individual across the top, our heterozygous individual down the side. We're going to populate this. This is looking very familiar. Okay, so in part A, big B, how many of those do we have? We have 1, right? So we can say 1 for big B. Our heterozygous individual here, there are 2 individuals out of 4 offspring. So we're going to have 2 of those. And then our homozygous recessive individual, we only have 1 of those, so we can put a 1 right there. And now we're looking for our phenotypic ratio. So this is just like what we did in question number 4. We have 3 individuals with that big B, so they are going to have the dominant phenotype. That is liking shiny objects, so we've got 3 there. Liking dull objects, that is the recessive trait, so you have to have 2 recessive alleles to show that. So we are only going to have 1 individual out of 4 that has that phenotype there, that homozygous recessive individual. All right, question number 6. Question number 6. Polydactyly is a genetic trait that results in the formation of extra fingers and toes. The allele for one form of polydactyly, capital P, is dominant to the allele for normal number of fingers and toes. So polydactyly is dominant here. Mr. Brown has this form of polydactyly, as did his father, but his mother did not. If Mr. Brown marries a quote-unquote normal woman, what proportion of their children is likely to have extra fingers and toes? Okay, so let's think about this. So Mr. Brown has polydactyly, so he has this dominant trait. So he could either be homozygous dominant or he could be heterozygous, right? Because either of these individuals is going to have the trait because they have the dominant allele. His father had polydactyly as well, so he could also be homozygous dominant or heterozygous there. But his mother did not. So his mother did not have polydactyly, so that is the recessive trait, so she has 2 little p's. Okay, so Mr. Brown, if we're thinking about Mr. Brown, his mother and his father are going to mix their alleles to get the combinations that are possible for Mr. Brown to have. So his father could donate big P's here, or he could donate a big P or a little p. But his mother, this is the important one, she can only give lowercase letters. She can only give these little p's. So this is her only option of alleles. So one of Mr. Brown's alleles has to be that little p. So this is Mr. Brown right here because he's getting 1 from dad and 1 from mom. This is the one he has to get from mom. He can't get a big P from mom because she doesn't have it. Okay, so we know Mr. Brown now. He is going to marry a quote-unquote normal woman. So that is going to be the homozygous recessive individual, right? They don't have polydactyly. So what proportion of their children is likely to have extra fingers and toes? How many of them are likely to have polydactyly? So we're going to do our Punnett square here. Mr. Brown going across the top, his wife going across the bottom. Be careful when you're using P's as letters. I don't actually like using P's as letters because it's really hard to see which one's big and which one's little. So just keep that in mind as you're using these. It might be helpful if this is confusing to you to maybe just pick a different letter. The letter doesn't actually matter. Just make sure you're keeping all of your letters straight. Which ones are big, which ones are little. So if we cross our individuals there, so half of our offspring have capital P's, half of our offspring are homozygous recessive, so these individuals are going to be normal here. These individuals have the form of polydactyly, so 50% of their children are likely to have this form of polydactyly, these extra fingers and toes, right? Half of those offspring there have that capital P. Okay, question number 7. I know this is a long video, but I think it's important that we go over each of these questions just so we know kind of what's going on here. So question number 7, we're watching the Jerry Springer show and they are trying to determine who the father of little Bobby Joe is. So Bobby Joe, he is type O, okay? So blood types are written a little bit differently. They're written as this I with a letter up at the top. It's just the way that you write blood types when we're doing these problems. So it looks a little different, but it's saying the same thing, right? Like one of his alleles has an O, the other one has an O, okay? Bobby Joe's mama is blood type O. So mom is also blood type O. And then we have 3 possible fathers here. Bubba, he is type AB. So type AB is going to have both A and B alleles there. Billy Ray is blood type B. So he could either be homozygous for B, or he could have one allele that's B and one allele that's O. And then Carl Jr. So Carl Jr. is our other potential father here. He is blood type A. So he could either be, just like Billy Ray, he could either have 2 alleles for A, or he could have 1 for A and 1 for O. Carl Jr.'s parents are in the audience, hollering that he is not the father, and they actually both have AB blood. So both of his parents are AB. All right? So you realize that they don't actually need to pay for DNA testing because we can tell them who the father is just by figuring out this with all these blood types. So we're trying to figure out who the father is here. So let's think about Bubba first. So if Bubba is the father, all of his children are either going to have A or they're going to have B. Those are the only alleles he can provide. Bobby Joe doesn't have A's or B's, so Bubba is not the father. Okay? But then we have Billy Ray and Carl Jr. here. It's possible that they're homozygous for these alleles, but it's also possible that they could be giving these A's and B's, or they could be giving these O's. So it's possible when we combine with mom here that this is the genotype we could be getting. But then we think about Carl Jr. with his parents. So his parents are AB, so they are giving either A's or B's. So if his parents are combining their alleles here, there is no possible way we could get this genotype, right? Because there are no O's involved. His parents are either giving an A or B. They don't have any O's to give. So he has to be this genotype. This is not possible for him to have. Because of that, we're kind of just left with Billy Ray here, right? If this is his genotype, that's his genotype. Let's put mom across the top here and Billy Ray down the side here. %50 of the offspring have a genotype that matches Bobby Joe. So congratulations, Billy Ray. You are the father. Okay. Question number 8. Let's do question number 8. Hemophilia is a sex-linked recessive disorder, so this has to do with the sex chromosomes, in which individuals cannot produce or produce in very small amounts coagulation factors that allow blood to clot. So we have a woman with hemophilia, and she is marrying a normal man. Okay? So this is sex-linked. It's going to be on the X chromosome, and it is a recessive disorder. So you have to have 2 copies of the recessive allele, or you have to have no copies of the dominant allele in order to show this trait. So these are situations where generally the males are more affected than the females because they only have that one X chromosome. So we have a woman with hemophilia. So I'm going to use little h's here to represent hemophilia. she is crossed with a normal man. So he is going to have a capital H because he does not have hemophilia, and he has a Y. So let's do our Punnett square and look at all of the possible combinations of alleles. So mom just has X's with little h's. Dad has an X with a big H, and he has a Y. So we can have a female who is a carrier for hemophilia, but doesn't have it. So those are our possible options across the top here. But then when we cross our X's and our Y's, all of the male individuals here looks like they would have hemophilia because they only have the little h, they don't have any big H's. So part A, can they produce a daughter with hemophilia? So these are all of our daughters, and none of them have hemophilia, so we can't have that. B, can they produce a normal daughter? Both of their daughters here are carriers for hemophilia, but they don't exhibit it. They are normal, so we can have that. Part C, we just answered. Can they have daughters that are carriers for hemophilia? That's what these daughters are, right? It's possible that they can give their offspring this allele. So yes, we do have daughters that are carriers. A son with hemophilia. All possible sons here are going to have hemophilia, so that's definitely a yes. And then is it possible for them to produce a normal son? And the answer to that is no, right? We have the only combinations of X's and Y's we have. We're getting our Y from dad, we're getting our X from mom. If mom can only give you a recessive allele, you are going to have it. So we can't have any normal sons, quote unquote, normal sons produced in this combination of alleles. All right, last page here, last couple of questions. Question number 9. Red-green color blindness is a sex-linked recessive disorder. So this is similar to what we were looking at with hemophilia. So we've got red-green color blindness. If a woman who is a carrier marries a man with normal vision, what genotypic and phenotypic ratios do we have? So I'm going to use big B's and little b's. I like using b's because the capital and the lowercase look very different. So we have a woman who's a carrier, so she has normal vision, she has the dominant trait, but one of her X's has a little b, has that recessive allele because she is carrying it. She doesn't exhibit it, but she's carrying it around. She is going to marry a man with normal vision, so he has a big B allele there. He does not have red-green color blindness. So what are the genotypic and phenotypic ratios? So let's draw our Punnett square. We've got mom with her alleles. We've got dad with his alleles. So we've got a daughter that has 2 big B's. We've got a daughter that is a carrier. We've got a son with normal vision. And then we have a son with color blindness. Okay, because they have that recessive allele and no dominant allele to overpower it. So our genotypic ratios here is just a 1 to 1 to 1 to 1 ratio, right? All of our genotypes are different. We have the daughter with 2 alleles for normal vision. We have the daughter who's a carrier. We have the son with normal vision. And then we have the son with red-green color blindness. Those are all of our different genotypes. And then our phenotypic ratios, we have 2 individuals that are normal vision females. We have 1 individual that is a normal vision male. And then we have 1 individual that is a colorblind male. So a 2 to 1 to 1 phenotypic ratio there. Okay. Question number 10. I'm going to go to a different sheet because this is a bit bigger of a problem than we've had before. Okay. Question number 10. So we've got 2 traits that we're looking at here. We've got mice that have round ears, which is dominant to pointy ears, and long tails, which is dominant to short tails. So 2 different traits that we're looking at. a mouse that is homozygous for both round ears and short tails. So homozygous for round ears, that's this one. And short tails, so that's our little t, is crossed with an individual that has homozygous for pointy ears, little e's, and long tails, big T, big T. So part A, what is the genotype of each parent? Well, we just answered that. That's right there. These are the genotypes of our parents. Homozygous for round ears and short tails, crossed with an individual that is homozygous for pointy ears and long tails. Part B, what genotypic and phenotypic ratios would you expect among the F1 offspring? So we're going to do a dihybrid cross with both of these traits here to figure out our genotypic and phenotypic ratios in our offspring. So this Punnett square is a little bit bigger. Instead of just 4 boxes, we're going to have 16 boxes in here. Right, because we've got lots more combinations of alleles to think about. Okay, so we're going to put our first parent across the top, our second parent across the bottom, and we're going to do all of the different combinations of alleles they could have. So we could either have this allele and this allele, so big E, little t. We could have this E with this little t. It's another one that looks the same. This second E could go with this little t. Or the second E could go with the second T. So even though we have 4 different combinations of alleles here, they all look the same. So this individual is going to be similar, right? All of the possible combinations of alleles, they only have little e's, they only have big T's, so those are going to be all of the combinations we can have. So all of these in here are going to look the same. They are all going to be heterozygous. Whoops, that's an A. They are all going to be heterozygous for both traits. They're going to have a big E, they're going to have a little e, they're going to have a big T and a little t. So the genotypic ratios, they all have this genotype right there. As far as our phenotypes, they are all going to have these big E's, which is round ears. They're going to have round ears. And they have this big T, which is long tails. They're all going to have long tails. All right? Part C, 2 of the F1 individuals are crossed. What phenotypic ratios are we going to observe among the offspring? Okay, so we have 2 heterozygous individuals that we're going to cross. So let's make our big Punnett square again. We're actually going to have to fill this one in because we've got all sorts of different combinations of alleles. All right, we've got our 16 boxes there. So possible combinations from this genotype, we can have a big T or a big E and a big T. We can have a big E and a little t. We can have a little e and a big T. And we can have a little e and a little t. Okay? This individual is going to be exactly the same because the genotype is exactly the same. So a big E, a big T, big E, little t, little e, big T, little e, little t. Right? So let's just populate this box here. Okay, so you want to be careful when you're filling this in, making sure that we're not missing any alleles. Okay, in our last row here, whenever you're writing these, the capital letters, if there is a capital letter, they always go first. So anytime we have a heterozygous individual, make sure you have that big letter first. Okay, so we've got lots of different combinations, lots of different genotypes, which means we might have lots of different phenotypes. So we're looking for the phenotypic ratios. So our possible phenotypic ratios, we could have round ears, and we could have long tails. It's possible we could have round ears and short tails. We could have pointy ears and long tails, or we could have pointy ears and short tails. So this is the way that I like to figure out my phenotypes, just kind of going through each box and putting a tally mark under these combinations of phenotypes. It helps me so I know that I'm not missing anything. So just to recap here, our round ears are the big E, pointy ears are the little e, long tails are big T's, short tails are little t's. So this individual right here is going to have round ears and long tails, so that's one for that group. Here we're going to have round ears and long tails. There's another one there. Round ears, long tails, round ears, long tails. Round ears, long tails, got a popular group here. Round ears, short tails, one for our next group. Round ears, long tails, round ears, short tails. Round ears, long tails, round ears, long tails, pointy ears, long tails, pointy ears, long tails. We've got round ears, long tails again. We've got round ears, short tails, pointy ears and long tails, and pointy ears and short tails. Okay, so if we tally all these up, we've got 9, 3, 3, and 1. So a 9 to 3 to 3 to 1 phenotypic ratio. So this is a really common phenotypic ratio that you'll see anytime there's a dihybrid cross, so crossing individuals that have two different traits. When we have heterozygous individuals for both traits, you're going to see this 9-3-3-1 phenotypic ratio. Okay, then there is one more question here. We'll go ahead and do the extra credit challenge. So in peas, green seeds, capital G, are dominant to yellow seeds, and brown seeds, capital R, are dominant to wrinkled seeds, little r. If a pea plant that is heterozygous for both traits, so big G, big R, little r, is crossed with a pea plant that produces yellow, wrinkled seeds, so this is going to be homozygous recessive for both traits, what proportion of the offspring would have green, wrinkled seeds? Okay, so we're going to have to do our dihybrid cross here again. So let's make our big Punnett square. Okay, so our first individual across the top, we could either have a big G and a big R. We could have a big G and a little r. We could have a little g, big R, or little g, little r. Okay? And then for our homozygous recessive individual, homozygous recessive for both traits, the only possible combinations is just a little g and a little r, right? So those are the only possible allele combinations that they can produce. So if we fill in this Punnett square, we don't actually have to fill it in all the way because all of these individuals are going to be the same if you go down the line, right? Because this is the same as this. So we're going to have a 25% chance of getting any of these possible genotypes, which results in 4 different combinations of phenotypes. So down here, the phenotype we're looking for is green, so that's a capital G, and wrinkled, 2 little r's. So we are looking for our green seeds, so that's not going to be this one, that's not going to be this one. So these are our 2 green seeds. And of these 2, this is the one that has wrinkled seeds, so this is going to be the genotype we're looking for. And because this accounts for a quarter of the possible offspring, we have 25% of offspring that have green wrinkled seeds. All right, so hopefully this was helpful for you guys. Please, please, please reach out if you have any questions about this. We're going to have our first live lecture on Monday at 8 a.m. Hopefully it runs smoothly. And then coming up at the end of next week, we're going to have our first chapter quiz. So keep that in mind as well, and start looking through the material and getting comfortable with the material. It's going to be on this chapter about genetics, Mendelian genetics, that the sub covered with you. So make sure that you understand that and get prepped for that chapter quiz. Again, please do reach out if you have any questions, and hopefully you guys are staying safe.